Right. Good morning, everyone. Most of you are here, right? Others will join. The thing is that there is a software issue, right? Nothing to do with that one. Sometimes when we start the meeting, right? What happens is the mic cannot be unmuted. Sampurna meeting even the machine should be restarted, right? Our machine gets restarted because Zoom getting issues, right? Those are the issues with Zoom. I'm on the platform, right? Take a hit to consumers right? And their platforms are not supporting for the things here and there. So I'm going to use the mic, right? I'm external microphone microphone gun they don't take it. Software right? Samaning Satir the Park usually twice a week, okay? Issue can orbit, right? Okay. Some of you are live, let's start, right? If you have any question, you can post them here. That's why it took, right? Time, right? No problem. So today we are going to finish about the organelles, so all the parts related to that one. In the previous class, I gave you the note up to the part we covered, right? So today I will give you the note from there onwards. Yes, today I can put it, right? So any question from the previous things? Any question before starting this one? From the previous things? Now I don't want to share this to the YouTube. It is already shared. So any question before starting today's session from the previous parts? Any more? Right? Campbell biology. Those who don't have the Campbell biology, microscope notes. Right? Practicals. Two practicals. Practicals. Identification test. It was a microscope. Right? Practicals. I just moved on to the theory part. Right? Microscope notes. Simple. Right? Okay. Practical. Right? So make it was a cell cycle. 11th edition, right? 11th edition, okay. Right? Small things are changed, but directly they have used this to. I cannot hear you if you speak. Message then if you really want to talk to me, I can love you to talk, right? Listen to me one thing, right? Now, most of the times, yeah, Campbell biology can be used, but the main thing, the book given by the department, right? It is based on Campbell biology. What has happened is small things are added and small things are removed, right? So in the marking scheme, you may have heard this word in the A levels, right? Marking scheme. We cannot write all the things that you know, right? So when you are writing the answers, past papers, right? Later they come into play, right? But somehow when you are writing, please stick to the points in the syllabus. Syllabus means the book, NIE book, right? You may know other points, okay, keep them in the mind. Don't try to write them. When you are writing them, sometimes you may forget the most important ones or things expected in the marking scheme. Then, select medicine. Sometimes new syllabus and all syllabus, there is something here. For you, it won't be affecting, right? New syllabus, all syllabus, because in the next year, there won't definitely be a new syllabus. New syllabus, so there won't be such thing, right? But new syllabus problems come into oh, spaces right? A, that is to paste some images, images okay. I pen of images, right? Might them printouts right? And then let's draw sets right? Color images and black and white images and drawing. Right? Next week when the travel restrictions are lifted, you can take the printouts, right? Can't put on printers, right? So as soon as you take the printouts, right? I'll tell you. Then yeah, okay. So you won't be having these syllabus problems, but somehow most of the students who have done very well, 3 years got the right? My students, right? Most of them have only followed this syllabus at the end, at the end, right? At the end, most of them have followed this syllabus. You can use the Campbell biology, obviously, right? Nice photographs are there, right? Electromagnetic, electron microscope images are there. Nice diagrams are prepared by them. You can get a very good idea. Even I use those diagrams for some descriptions. Today also I'm going to show on microtube view, right? But when you're writing the answers in the final one, the bulk in the Campbell biology is so high. The Campbell biology right? Keep that as an extra reading book. Extra reading book, right? Right? You write the things in the Campbell biology, right? So so okay, if you can write all, but we all are human, we forget one or two. Textbook in the textbook, oh sorry, syllabus points halila, you drop the points in the syllabus and you write some points in the Campbell biology. Unfortunately, most of them are not in the marking scheme. So there is a problem at the end, right? So don't study the Campbell biology, just read it, just like an extra reading book, right? What you are going to do is, okay, I saw the camera thing, right? So what message that right? What you are going to do is following the syllabus, right? Campbell biology 11th edition, it's okay, right? Take only one edition. If you have the 10th edition, don't buy the 11th one. They have used the 10th edition to write your syllabus because it was written early in 2017. Right? 2017, so they used the 10th edition. 19 17 right? So they use the 10th edition. Now new editions are there. Even if you have the 9th edition, you can hard copy lazy, right? Hard copy bad. It is not soft copy. Even in my theory classes, okay, CD print that is how we do, right? So keep it as an extra book if you have, right? Group meeting link group right, so that is what I want to tell you, right? Oh yeah. Have a Campbell right? Campbell, some things of the Campbell is drop, some things of the Campbell is drop, right? Environmental biology parts are Sri Lanka. Environment we talk about the Sri Lankan environment. Those parts are added in the lessons. You will see, right? Some lessons are totally from other sources. Other sources I'll give you those sources also, but Sri Lankan things are not in Campbell biology, right? The environmental biology parts, especially, they are there, right? So not 100% Campbell biology, but most of the things are Campbell biology, right? Okay. Now let's go to the syllabus. Sorry, book, right? Any other questions? Questions? No. No questions, right? Now we are going to talk about the, if you have any question, please put it, right? Okay. Cytoskeleton, right? Cytoskeleton means when you talk about the cytoplasm, right? When you talk about the cytoplasm, right? In both plant and animal cells, the area surrounded by the plasma membrane, right? Now, even when I'm giving you the 3D example, I asked you to remember that one as a balloon filled with water. So this is the balloon membrane, just like the cell plasma membrane, and water is just like the cytoplasm, right? But it is not exactly like that. It is just like this, right? Now, yes, there is a balloon filled with, imagine there's a balloon filled with water. You are going to enter or you are going to insert some fibers, right? Coir fibers, right? Or some plastic fibers here and there. You're going to insert. So these fibers will be like this. They will be spread like this everywhere, right? Likewise, inside the cytoplasm, it is not just a gelatin structure or it is not just a liquid structure. Messages right? You can message me, no problem. Right? Group right. Then right. So inside the cytoplasm, right, we can find similar structure. So it is not just a liquid. It's not just a liquid, right? Microtubules, that means small tubes. Microtubules means tubes, tubes, larger one. Tubules means small tubes. Microtubules means very, very, very. It's correct, right? Cell is very small. Inside that one, even inside the cell, you can pay 5 tube here. Very, very, very small being, right? Microtubules, right? And some filament-like structures, right? So all of them get together and forms a three-dimensional structure. All of them get together and forms a three-dimensional structure. So it is called the cytoskeleton. Right? So what would be the use of this cytoskeleton? What is the use of having such thing? First, for us, it is really useful, right? That means animals. We don't have a cell wall. So to maintain the shape maintain these things are really important. They are not very rigid like plant cell walls, but to maintain the shape, it is important. First thing, right? And now, when we were drawing, we have drawn like nucleus, right? Chloroplasts here and the mitochondria, like this. So actually, these organelles are embedded. They are sunken in. They are embedded in different places, at specific places in this cytoskeleton. I'll show you a 3D image, right? They are embedded. So they stick in one place. Otherwise, what will happen? All the chloroplast right? When you are standing like this, right? Due to the gravity, all the organelles will come down. That can happen. If this is a liquid, imagine the balloon situation. The balloon, right? Water-filled balloon. When we hang it like this, if some things were floating like this, all the things will be deposited here. All the things will come down with the gravity. Likewise, when you're standing like this, all the organelles will go down. If not, right? So it is prevented by this structure. Imagine there's a structure like this. Right? It holds the organelles like this. Really good, right? So first thing, gives a shape, right? To maintain the shape, right? Second thing, organelles are kept in specific places. Third thing, right? This cytoplasm circulates. Circulated, specific name is there, name right? This is not a solid, or this is not a constant way of just cemented or something like that, right? This rotates, right? It rotates. Imagine this just like a liquid just like stirring a liquid. Right? This can move. Sometimes even organelles can slowly move, right? This facilitates these movements inside the cell. Right? The cell like actually movements, small movements, right? This structure facilitates these movements inside the cell, right? That is called cytoskeleton, right? Text mainly we follow the text, and other than that we follow the other books or resources, right? Okay. අනිත් එකත් මට දාන්න පුළුවන් රේවන් ජොන්සන් එකත් මම දාන්න රේවන් ජොන්සන් එකත් right නිකන් තියාගන්න ඔයගොල්ලෝ අහනවා මගේ ඒකයි it's good right no problem you can ask anything රේවන් ජොන්සන් එකේ soft copy එක තියෙනවා but රේවන් ජොන්සන් is not used to write this one right පාවිච්චි කරලා නැහැ රේවන් ජොන්සන් එකත් මම දාන්න right cytoskeleton is the supporting structure of the question yeah Raven Joseph same thing, right? Nucleus, not nucleus, but the points that are taken are directly taken from this one, right? Cytoskeleton is the supporting structure of the cell and multi-game multi-game eventuals and you should go in the same page, right? It's okay finding all the resources and all that. Together eventuals green biology green biology the right book green green biology but ani todaslo matagathne puthe nam matagathne red color book ikkada then goda use cheyenne mane ek images diagrams karna we take the diagrams from that one red color book mokadda diga namak thiyena right usually red book kiyala kiyana natta green biology right ekkenek thamai ROPR green har mokkari hom kiyala green person right a green biology book Campbell biology medicines right but now I වැඩ කරන්නේ him බෑ so main thing is පොත් ඔක්කොම දිහා බලලා බය වෙන්නත් එපා කොට කැම්බල් එක රීඩ් කරන්නද සමහර ළමයි කියනවා මම කැම්බල් එකත් කියවනවා green biology එකත් කියවනවා රේවන් ජොන්සන් එකත් කියවනවා මමවත් ඔච්චර කියවන්නේ හරි මේක කියවන්න syllabus එක main thing is syllabus right බය වෙන්න එපා අන්තිම කාලේ මට කියවන්න බැරුණා don't go right if you have the time now finish them please stick to this right even physics chemistry they to කියයි මේ පොතේ තම ප්‍රශ්න හදන yes there are some books for physics and chemistry most of the questions are directly taken from these those books there are thousands of questions for one lesson එකකට 1000ක් විතර එහෙම කරන්න බෑ so what you should do in those especially in physics right ඔය තියෙනවා කියයි මේ පොතෙන් තව ප්‍රශ්න හදන්න මේ පොතම හදන්න එහෙම කරන්න බෑලා මේ වැඩකුත් no need for the by hearting of the questions right learn the theories very well theory and application හොඳට ඉගෙන ගන්න එච්චරයි ඕන question එක handle කරන්න right cytoskeleton is a supporting structure of the cell and maintains shape due to this structure otherwise just like a balloon filled with water it's a problem especially in us animals right it is more important for animal cells which lack cell wall right cytoskeleton is made out of microtubules and protein filaments later we are going to learn microtubules means tubes again tubes very 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 small micro tubes are small the small part of tube is called tubules again if this is a tube tubules microtubules very small tubes additionally it is dynamic changes right hence has the ability to break and reform as needed yes and it is not a rigid structure just like our skeleton to break and reform we cannot do that right but this is such thing can be when you are pressing like this when you are pressing see pressing the muscles massage karna somewhat right and in most of the cellular activities this breaks this structure breaks right and reforms like this right okay main parts are microtubules give me 1 second I will show you one image of that one right so idea ki මම හරි බයයි මේ software එක දාන්න මේ ඕකෙන් තමයි උදේ හිරවුනේ අපරක් ඉන්න I have prepared some images for you especially green biology images sorry I have prepared images of the Campbell biology ඒක load කරනකොට උදේ ප්‍රශ්නේ ආවා right I'll show right two images are there one is 3D one the other one is not a 3D one you can get it right අපරක් ඉන්න Right. Within. Now I hope you can see this one. See, the organelles are sunken in. They are sunken. They are tightly fitted. The mitochondria is tightly fit. Right. So this is the skeleton everywhere. Right. Cell membrane is here. Right. ER and ER endoplasmic reticulum is the purple one. It is also tightly fitted. Ribosomes are there and also microtubules. Let's learn them later, but I won't tell you how it is tightly fitted. Right. So everywhere it is mesh-like. Cytoskeleton all together, right? So whole structure we call it cytoskeleton. Chromi images right? See now, yeah. There we are, theory part, right? Let's discuss them one by one. And the images right? Microtubules. Right. Mama image dana, you will not see me, right? Full I'll put an image and I will read it, right? And I will read it. Campbell biology image, right? I'll put the image so you will get a good idea. Right? I'll put an image and let's discuss with that one, right? They load karana the images. Tamat by load karana meeting again. Right. Okay, you will not see me, right? But you will see the images. So I can easily explain, right? Then I will draw in the book, right? Let's read it. Pick you. Hollow tube. So microtubules. This structure is formed from three things, right? In this structure, three main structures are there. That means in the cytoskeleton, right? Three main structures. That means to build the cytoskeleton, three things participate: microtubules, microfilaments, and intermediate filaments. Right? Let's learn them one by one. First one, microtubules. Hollow tubes. Hollow tubes means they're inside that there is a hole. Right? Hollow tubes. Wall consists of 13 columns of tubulin molecules. 13 columns of tubulin molecules. I'll draw you and tell you, but let's try to identify it here. It's nicely given. Right. This is one column. 13 right? This is one column. 13 columns, right? 13 columns. Right. 13 columns. Protein subunits. Subunits from tubulin. Protein called tubulin. Actually, it's a protein. Since it is forming tubes, we call it tubulin, right? Main functions: maintenance of cell shape, cell motility. Right? Motility means, right, moving, motions, right? Motility and mortality. Mortality means death. Mortality, right? This is motility. As in cilia and flagella, right? Chromosome movements in cell division and organelle movements, right? Look at the image again. Image I'm going to, yeah, right? This one. Those who don't have the Campbell in a theater, right? Then right? Right. Listen to me. Right? It's a hollow tube. Yes, hollow tube. That means in the center there is a hole. The next word of right? In the Campbell, lot of other stories are there. Take what is necessary, right? Wall consists of 13 columns of tubular molecules. That is something like this. Tubular molecules means that when you are drawing, we draw the wall like this. Right? Wall. This is the wall, right? The outer wall. Now we are going to talk about this wall, right? When we take this wall, it is consisting of 13. Let's look from this side. That means this one, right? It consists of 13 columns. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. That means 13 protein molecules can be formed. They run as columns. So make a column. This is one. Actually, one column. This is the second one, right? I'll take another column, second column, right? Yeah? This is the second one. Second one. Okay, this is the third one. The third one. Likewise, 4, 5, 6, 13 columns, right? So if this is one column, 13 such columns get together and form. In one column is not a constant one. It composes of small protein molecules. That is what it says. The wall consists of 13 columns of tubulin molecules. So this is protein. Protein, right? Tubulin protein. Right? This is one column, so it runs like this. Piles you're going to take small pebbles. Pebbles glue pebbles, glue. One column of pebbles, right? Pebbles. And another column. Glue. Right? Second column. And what you're going to do is you're going to keep all these columns together, 13 columns together. Right? Take pebbles like this and glue them together. So one column. And you're going to take another, likewise 13 columns are formed, and all the columns are then glued in a way that it forms a tube. That is called 13 tubular proteins, right? Okay. Sometimes students, at the middle of them is a hollow structure. It's a hole, mukutne, right? Empty. I'll show you the image again after this one, right? Is it clear for you? So the protein subunits, these are called protein subunits. So this one, I can draw it in 3D. The subunits run like this. Lesson in never project. Right? This is a one column. Right? The other column. Likewise, there are right? Columns. 13 columns. Yeah, right? Last of right? 13 columns and then the other columns. Right? They run like this. That's separate, right? At the middle, there's a hole. Middle, nothing. Other than we have to do that. Very, very small hole, right? Hollow structure means that one, right? Hollow tubes. This protein single unit is called tubulin. Right? What is the function? Main function? Maintenance of this cell shape, right? Maintenance of the cell shape. There is one function. They have not considered about the punctuation marks here. Mohut punctuation marks yeah. Cell shape. So in the cytoskeleton, it helps to maintain the cell shape. One thing. Second thing, cell motility. Cell motility in the Campbell biology lesson diagrams, right? Motility means for the movements of the cells, especially spermatic. Right? In the sperm, cilia and flagella. Right? Cells move using the tail, cilia and flagella. Later we are going to discuss separate, right? For this one, and inside the movements, right? Motions, movements of the cell, right? Cell motility, cilia flagella, right? I'll tell you the chromosome movements, actually that is in the next part, cell cycle, right? Chromosomal movements in the cell division and organelle movements. Organelle movements here, organelles are moved inside the cell. Organelles move when it is necessary, right? Inside the cell using the chromosomal movements means I just tell you. Now, when you talk about the mitosis and meiosis, we discussed the parental chromosomes separate 23, 23. How it is separated 23, 23, right? Structures have been lesson, right? Chromosomes, they are binding and forms a nice structure called aster. Aster, right? Here microtubules participate, right? They crack them, right? And separate them. for the separations of chromosomes, these tubes, the tubes join, right? Right. Let's talk about that. A new topic, right? Okay. I think that is clear for you. Clear is? One moment. Right. Maybe method. I don't know whether you can identify. Right. Center is a hole. No? Center is a hole. Center is a hole. Center is a hole. So, right. So wall. We wall right. These are columns. Right. Let's go for the next one. Like right. Two intertwined strands of actin. Each strand is a polymer of actin subunits. Actin killing, right? Two intertwined strands of intertwined strands, right? That is super easy. It's not that complicated, right? Now imagine again, there's a long strand. There's a long strand like this. 1, 2, 3, long strand. Right? There's another long strand. Now they are intertwining. They are intertwining. Just like DNA, not in the similar manner. They are intertwining. These subunits, subunits. This is a strand. It's a subunits. They are actin. We all have these things, especially now muscle cells. Muscle cells, right? This is how the muscle mechanism works actually. Actin and myosin, right? Or Huxley's mechanism here. When you are drawing, if you can remember O level, right? O level muscle cells, when you are drawing, not the smooth muscle cells, cardiac and skeletal muscle cells. Striations are present here when we will you have drawn. Striations are present. Those are these fibers, right? The fibers. Huxley's mechanism of muscle movement, right? It's a topic for us later. Right. So somehow these are present in muscle cells a lot, right? So the second type is actin protein is present like this, microfilaments, right? Microfilaments. Like this, and there are two intertwined chains. That's all. Very simple, right? Here not that. Two intertwining strands of actin. Each strand is a polymer of actin subunits. Right? Actin is the protein. Maintenance of cell shape. Maintenance of cell shape. Skeleton right. Tension bearing elements. Changes in shape. Muscle contraction. Muscle contraction. Right? Here it is given inside brackets because officially we have to discuss it later, right? Cytoplasmic strengthening, plant cells, cell motility, as in pseudophodia. Can we make up, right? Pseudophodia later, you know. Practice. Pseudophodia in amoeba. Amoeba level. How amoeba moves, amoeba doesn't have any specific organs or whatever. When it wants to move and when it wants to take food, it forms false feet. It is called pseudophodia in Latin. These false feet are formed by these structures. Right? Right? False feet. This pseudophodia. This structure that's why I have drawn, right? Actin, myosin, the muscles we discussed it, right? Pseudophodia. Food particle is here. Then this surrounds the food particle. Right? Food particles will surround color. Right? Cell eating process. We call it cell eating. I search search phagocytosis. Phagocytosis, right? Possible. Right. Cell eating process. So those are the functions, right? Cytoplasmic strengthening, plant cells, cell division in animal cells, cleavage, furrow formation in the cell division. Then I kill them, right? Okay. Next one, intermediate filaments. These are microfilaments or actin filaments we call them. These are intermediate filaments. Fibrous proteins supercoiled into thicker cables, right? One of several different proteins, example keratin, depending on the cell type, maintaining of cell shape, again functions, right? So all three subunits or subparts, microtubules, microfilaments, intermediate filaments, all three supported or participate to maintain the cell shape, right? Then anchorage of nucleus and certain other organelles in the formation of anchorage of nucleus and certain other organelles, that is anchorage can support, right? There is formation of nuclear lamina. Can you remember nuclear lamina? Right? Nuclear lamina. Immediately after the nuclear envelope, nuclear envelope, if this is the nuclear envelope, two membrane on Q. We cannot draw it inside this. The nuclear lamina. This provides the anchorage support. Nuclear, it's a very small one. I have purposely drawn it very large. The nuclear lamina support. Imagine the roof is the wall, right? To nuclear envelope. You are holding that like this. Then you are like that. Right. The best example for this, this just like a rope. Rope, how a rope is consisted of, right? Small threads are there. Two threads, lot of small threads are nicely intertwined, tightly intertwined. Rope, right? Likewise, small fibers are tightly intertwined, lot of fibers, right? These proteins, previously we told, tubulin, actin, but here, depending on the cell, the protein may change. For an example, keratin sometimes, right? May protein, right? So, a lot of fibers get together and winds nicely, tightly, and forms a rope-like structure. Right? That is called intermediate tubule. Right. For the last time, I'm going to show you the three diagrams again. But right. Protein columns together, right? Two strands got together, and this one, keratin proteins. See how fibrous they are. Just like a rope. I hope you don't need to remember these diameters, diameters only. But remember the size. My thing. Intermediate means size is intermediate in between them. Fibers 25 nanometers, right? Oh, sorry, microtubules, right? Then microfilaments, smallest 7 nanometers. Then intermediate can in between them size, right? 8 to 12. That is how the names come. Right. Any question? Any question? So the functions, right? All three cytoskeleton functions. Cytoskeleton is formed from these three, right? What are they? Microtubules, right? Second one, microfilaments. They are everywhere, coming like this. Microfilaments and intermediate filaments, right? Intermediate filaments. Right. So we learned about all three, right? Functions. Right. All together, the cytoskeleton functions. Provide strength to the cytoplasm. Do you agree with me? Provide strength to the cytoplasm. Even though physical classes, but then since we have the facilities, we can do this. Right. right? Provide strength, yes, obviously. Strength provide. Being like this provides the strength to the cytoplasm. Right? Next function, encourage organelles and cytosolic enzymes, right, of the cells. Cytosolic enzymes now. The term encourage means holds the organelles. This one. ER is everywhere, so holds them, anchors, right? Encourage. Movement of cytoplasm, cytoplasmic streaming. Cytoplasmic streaming, that means cytoplasm circles. What area? Right? Stir just like stirring something, streaming. Right? Position organelles and move chromosomes when necessary. Chromosomes move in the cell division. Right? They are moved in cell division, but all the things are moved. Right? Cytoplasmic streaming. Maintain the shape of the cell, mainly in animal cells. Right, those are the functions. Did you understand? Because took a lot of effort because we didn't discuss at least a single word in all levels. Right? Totally new. Okay. Is it clear for you? Not others, all of you, them, new students, old students. This is a different process, different approach for both. Calling Lama in online, and I know their level, but this is the totally different level, right? So all of you should respond, right? Then I only I will get some confidence. Right? Now, when we finish this part, we finish majority of the first lesson. Right? The others didn't get Terunet. Respond to me. Communication done responses. Take some time if you want, 1 minute. Tell me. Individually communicate. Right? Send me, you understood. Even the 25 students, 24 of them are saying understood. If someone don't understand, just don't say because others are saying, right? I didn't understand. Right. I thought at least to do something. Julia understood the I'm confident, right? Okay. But finish. Okay, majority of you have understood. Anita done, right? It's really important to have the feedback of you, right? They have not because this is a really debate. O levels for textbook, right? They are not really difficult theories. They pay on it. These are microtubules. And a lot of misleading information is there, right? If you just Google it up, a lot of misleading and different than what is essential for us. A lot of things are like the medicine level, right? Psychological level, psychological, right? Different things are there, right? Then we are moving to the cilia and flagella. You already know about cilia and flagella. I tell you where you know them and how you should know them. Right? Cilia and flagella we met in different places. They are like this. Paramecium, microorganism, hair-like structures. They were important for their locomotion. Cilia. Cilia are short and large in number. Right? Numerous kilometers, right? Large in number. That means the amount is high. Flagella is long. In bacteria, you identify flagella, and in euglena, long and most of times 2 or 1. Limited, right? It's called flagella. When it comes to our body, we find cilia. Cilia, right? Ciliated epithelials. Officially, we have discussed here. Can you remember or can you tell me if actually think about O-level thing, right? In one system, if someone can remember, tell me. Ciliated epithelium, right? Okay. So ciliated epithelium. Where did you find that, right? So these structures, cilia and flagella, shares the common structure. Only the length is different. The structure, composition, the way they have formed is similar, right? Cilia and flagella. So we are going to learn, really important, right? Yeah, very good. Digestive system. There was epithelial issues. Ciliated epithelial was, yes, in the respiratory system. Right? And iten you going to female reproductive system ciliated epithelials are there, right? To just push the egg, right? In A levels we learn, in O levels we didn't learn that one. ඔයගොල්ලෝ ඉගෙන ගත්තේ fimbria වලින් ඇතුළට ගන්නවා කියලා විතරයි. Only fimbria takes them in. That's all you learn, right? Cilia and flagella share a common structure. Flagella are long, elongated structures. This is present in euglena, bacteria, flagella, right? Structures. And cilia are short cellular projections that are often organized in rows. So, nothing in rows here. Best example, cilia, your toothbrush. Negative fibers, right? Cilia like that. Toothbrush, lines. Right? Cilia are more numerous. That means higher in number, higher in the quantity. Cilia are more numerous. Like imagine this is in 3D. 3D වලින් හිතන්න. Panadol එක ලොකු එකක් structure එක. මේවා. මෙයා හිතන්න. Paramecium කියලා. So this is totally covered. Totally covered in cilia. මේක line එකක් නෙමෙයි. It's 2D, right? This is totally covered in cilia. So a lot of cilia is present. Cilia are more numerous than flagella on cell surface. They are made of microtubules. They are microtubules are like flagella cilia we have discussed. So 13 rows, right? Tube, tube, 13 rows of protein. The microtubules, right? Okay. With 9+2 structure, this is important. 9+2 structure. 9+2 means like this, right? Okay. Now this is the flagella or cilia, right? We are going to cut it cross section. Cilia cross section. Cut. When you cut that one, you will find something like this, right? If you cut it from here, cross cut, right? AA dash, cross cut. 9+2 means 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 doublets. Right? 1, 2, 3, 4, 5, 6, 7, 8, 9. 1, 2, 3, 4, 5, 6, 7, 8, 9. Right. 9 doublets. Can you? Okay, 9, right? 9 structures. And in the middle, there's a couple. It is 9 plus 2. All these single ribs are microtubules, which we learned earlier. Microtubules, right? It's like this, and this is covered, surrounded by membrane, which is exactly similar, which resembles plasma membrane, right? Mea membrane is covered. Is it clear for you? 9+2. Then one more thing, right? When this comes here, then what a base second. This joins with a base. That means if this is the flagella, this should be connected to a base. Join in the back. Here. Here. Right? When it is joining, it shows a different structure. Earlier this one, and this base. Base means different here, right? It's a nice structure. It's a modification of this one. So I'm going to modify this. It joins with the tubes here. Rope is there. Right? You can rope projections. Projections of a rope. If I want to join this, I can join this one with the projections which flies here. See, right? Right? 2 is here, surfaces. I want to join my left and right hands, right? I have two structures here. Right hand has one structure. I get them together and fix together. Now, left and right hand is nicely fixed, tightly fitted. Right? Again, if I separate, again if I join, separate like this, right? So, one more tubules join. Where does this rise? This is coming from the base. Will I have the space? Continuously, right? Then you will see. Now there are 9 triplets. And this disappears. At the base, 9 triplets. This red color one is something emerges from here and joins or binds with the previous one, right? So at the base means where this cilia or flagella joins that particular surface, we can find 9 plus 0 structure. Here is 0. 9 plus 0. Right? Unmuted. They have it. Yeah. Okay. Did you hear that one? Harald? හරියන්නේ මොකද අයින් කරගන්න. ආයෙත් එහෙනම් ready. දැන් අයින් කරනවා. ආයෙ එන්නම් බැරි වෙන අයින් කරන්න පුළුවන්. එතකොට ඉතින් හරියන්නේ. Any question? Is it a question or something? Yeah, right. So this is the structure, right? Is it clear for you? Any question with that? They are made of microtubules with a 9 plus 2 structure. Previous one, think only about the black dots, right? 9 doublets, couples, 9, right? Of microtubules are arranged in a ring with 2 single microtubules in the center, 9 plus 2. They are covered by a plasma membrane, which is exactly similar to the plasma membrane, right? And bound to a basal body. Basal body means to a particular surface right? Which anchors the cilium or flagellum to the cell. The basal body has 9 plus 0 arrangement. No microtubules in the center. No microtubules in the center, but 3. Right? Functions, right? Act as locomotor appendages. Locomotor appendages means just like I told you, locomotion. Locomotion. But මේගොල්ලෝගේ cilia flagella වෙනස්, right? Eukaryotes ලගේ prokaryotes ලගේ වෙනස් වෙනවා. So bacteria cilia flagellum is different from this one, right? Sperm එකේ හිතන්න from the sperm, right? Can move fluid over the surface of tissues. This is where respiratory system comes into play. In the respiratory system, what is the fluid secreted in the respiratory system? O-level knowledge. It's really easy for me if you have the O-level knowledge. Very lazy, man. Chemical biology, right? Yeah, very good. Mucus. I think right. You're working good. So in the epithelial like this, right? Name is ciliated epithelium. So cilia is present here. On that, secretory cells secrete a liquid called mucus. Even now, if you clear your nose, you will find that mucus, right? Liquid. So that liquid is moved. Liquids move. Right? Motion of liquids. Right? Yes, good. Can find. Good. Next one, fallopian tube. Right? Inner lining of oviducts help to move an egg towards the uterus. Right? In the reproductive system, we have discussed. Don't draw them here. In the respiratory systems, we have to discuss, right? Oviducts. Only one side. Eggs are released. So here fimbria is here, fimbria, right? When the egg is released, it should be pushed. It is pushed again in this inner layer. Okay. This inner layer. There is an epithelium. It is also ciliated epithelium. Right? So when an egg comes here, it is pushed, right? And brought towards the uterus. Outcome is pushed, right? Using this ciliate. Epithelials is a huge topic here, right? Later we have to discuss. Right. So, me Campbell again, me something taken from the Campbell. Make it right? Make it right. Is it clear for you? Should be clear. Right. Next, yeah. Next, centrioles. Really important. You have not even with TIPA. Name was there in the O-level science book, but you didn't discuss anything, right? Now, up to this point, you have discussed about organelles or structures which present only in plant cells. Only in plant cells. Animal cells, we don't find it, right? This is the only structure that you learn which is present only in animal cells, right? Centrioles. Centrioles especially participate for the next thing that we discuss actually, right? That is the cell division. In the cell division, everything is managed. You're doing an event. Coordinate one or two coordinators. Coordinate. Everything is coordinated, right? Everybody meets. Principal coordinate, right? Organizing, organizer, coordinator, different names are there. Organize. Main organizer, right? Everything, right? Especially the chromosomes organized. You should go there, everything. We are discussing later, right? So centrioles are present. Centrioles are again formed of microtubules, right? Okay, let's discuss and write it. Two, perpendicular, right? Perpendicular. Two, right? Two centrioles. They are located perpendicularly. So when you are cutting that one, when you are taking a cross section of a cell, what you find is something like this. Chi. The other one is cycle. Like this, right? Right. Centriole is made up of cylindrically arranged microtubules. Again, microtubules, right? Which are non-membrane bounded. Previously, in cilia and flagella, they were covered with a membrane, but this is not covered with a membrane, right? Membrane bound subcellular components present only in animal cells. See? This is the only thing that we learned, present only in animal cells. Each centriole composed of 9 sets of triplet microtubules arranged in a ring of 9 plus 0. Just like the previous structure, 9 plus 0 structure, right? Triplets arranged in 9 plus 0, right? That means nothing to draw again. Likewise, 9, right? Center, there is no such thing, right? A pair of centrioles which arrange perpendicular, right? Perpendicular to each other are located in the region called centrosome near the nucleus. Nucleus around the nucleus. Because why it is near the nucleus? In the cell division, chromosomes is the important, chromosomes is the most important thing. So it can very easily attach with the chromosome. Later, right? That's why it is near the nucleus. So every time you will find this structure near the nucleus, even in O-level book, O-level cell, right? O-level cell, O-level cell. O-level cell in green biology, right? Do we have the O-level cell? Should be O-level. Mama, I'm going to go. All right. Balance centrioles with the vein. Nothing transparent image. I put a transparent image. Near this, right? See? Near the nucleus, right? Near the nucleus, you can find the cell, right? Near the nucleus, 2 centrioles. That's the banana, right? But it was not mentioned a lot. Right? Is it clear? Functions produce aster structure class, right? Aster last structure in the cell division we are going to discuss, right? Yeah. Right? Aster. Right? Aster. You don't even know the single names for these things. I mean, your single number to right? I don't think that Aster, I think Aster, right? In Sinhala, there is a name, right? And I don't know about the Tamil names, right? Produce aster and spindle in cell division, right? Okay. Then next one, central vacuole. Central vacuole. You know about the central vacuole, right? Nothing to discuss. Large central vacuoles are present in plants, but even animals have small vacuoles, right? So vacuole is again just like a small balloon now. Small balloon inside the large balloon, right? It is like this, right? Especially in plants, this is the stores. Right? It stores mainly water, right? Mainly water is stored. Vitamins, ions, pigments. Pigments means whatever chemicals which can give a color, right? Are stored here. So this is covered by again a membrane called tonoplast. Right? Tonoplast. It also resembles the cell membrane. Cell membrane resembles curve, right? Central vacuole is a large structure bound by tonoplast, bound by a membrane. Membrane is called tonoplast. Right? Filled with liquid called cell sap. So this is tonoplast. Cell sap. Both of them together we call vacuole. Right? The composition called cell sap found in plant cells, most of them. Right? The composition of sap differs from cytosol and it contains water. A cytosol means the outer one. Now in cytosol, there are organelles, but there are no organelles inside this one. This is also an organelle, right? But if you take a plant cell you have got to large majority of them is from this one. Majority, right? transparent transparent right transparent annual එක see large area is covered by this one, right? Comparatively large area is covered by this one, right? In O levels, nothing. Good level covered. The composition differs, right? So it contains water. Majority is water. Majority is water. Ions such as potassium and chloride, and sometimes water-soluble colored pigments. Colored pigments. Chemical pigments. They got done now, right? Chlorophyll. Later we are going to learn, right? Chlorophyll, carotene, xanthophyll. 4 pigments, right? Chlorophyll. They got chlorophyll A and chlorophyll B. 4 pigments. In our skin, melanin pigment. In our red blood cells, hemoglobin. Those are the examples, right? But here, chlorophyll and several pigments. And anthocyanins such as anthocyanin, carotene, xanthophyll, right? Leaf color right? Anthocyanin is red color. Anthocyanin. Some leaves are red in color. That is due to anthocyanin. Right? And anthocyanin red, brown, it depends on the structure. Red, not this red. Reddish brown. Functions. Stores water and other materials such as sugar. Stores. This is mainly store. That was store. Right? Ions and pigments. Maintains water balance of the cell. Yes, this maintains the water balance. That means if the water amount in the cell is less, if it wants the water, this fills water. There's a nice term called turgidity. Turgidity. Turgidity, right? Some cells, right? Some cells are very fragile, even the plant cells, right? So they go plants balsam, balsam plants. Immediately, if they are removed from the soil, if they are uprooted, what happens is they start to wither, right? That is due to the loss of water from this tonoplast, right? So they also supports the mechanical, right? Mechanical support, right? Maintains water balance of the cell, water balance, right? Gives turgidity and support to cell. Later we have to discuss underwater relationships, right? Turgidity means fill karga, right? Imagine a balloon but if you fill that with air, then that becomes like this. Turgidity, filling water inside and becoming rigid. Right? Immediately we uproot that one. What happens is water gets vaporized and they become like this. Produce colors in some plants with sap pigments. These pigments present in that one, just like I told you, in some, especially in begonia. Begonia, color is given to the cell and ultimately to the structure by this. Stores soluble substances needed for cellular activities. Yes, later, cake activities to only respiration, even for the respiration, some things are stored, right? Okay. With that, we finish that part, and I give you a small interval now. And then let's talk about the extracellular component. Cell wall is finished. I came like that. Cell wall, plasma membrane likewise. So we have to discuss another very important one today. Cell junctions, right? Cell junctions are important. It's a difficult thing, right? Extracellular matrix, it's nothing. Right? Okay. So if you have any question, you can post them to the chat here. Right? Take a small break. Let's join in 10 minutes, right? Let's join in 10 minutes. Right? If it is clear for you, everything you got done, right? Everything is clear for me. right. Samara power cut to name. Okay. I will send the recording for them, right? That space, you can table the text speaking. In the student guiding, right? There's a text, right? Table fill the difference between eukaryotes and prokaryotes. We discuss back to the discuss somewhere around 1 hour, right? Fill that, right? We tube through, right? We are going to quickly go through the tube to note, right? Note again. Gaps to go fill kerala, mummy. Some of the students are, yeah, then we must right? Immediately after finishing all of them, I'm going to go through that one quickly, right? So blanks of fill and I'm going to give you the printouts. Printouts gun, right? It was very correct, right? Right. Connect yes. One of them have power cut, so he or she will not come back to me. Then cell wall we finished. Cell wall finished kara. Extracellular matrix. Extracellular matrix means now when we talk about the animal cell, you know that there is no extra outer covering. It's like for the plant cell, right? So what you feel is the animal cell is very exposed. Animal cell is exposed, right? But it is not, right? There is a part called extracellular matrix, right? Nice diagrams are in the Campbell balance. Right? So I'm going to show that to you. If this is our cell plasma membrane, right? Extracellular matrix. Name itself says, right? These are tubules. Like this. These are tubules, right? Now, extracellular matrix. That means beyond the cells, there is a matrix, right? So this protects the cell somewhat. We can protect cell, right? This is not just exactly similar to a cell wall, but this protects, provides some rigidity to the animal cell. Right? When you were small in O levels, what did you think about animal cell? Now what did you learn? Right. There are a lot more other things to learn also. Right? This becomes advanced. So inside this cytoskeleton membrane, outside this one, extracellular matrix, right? Although animal cells lack cell walls, they do have elaborate extracellular matrix, ECM, right? Main components of ECM are glycoproteins. Glycoprotein. What is a glycoprotein? Now, glycogen plus proteins. Where did we meet them? In the cell membrane. Can you remember? Proteins. Carbohydrate bound to the proteins. Right? When carbohydrates were bound to this one, I call them protein plus carbohydrate is glycogen. So glycoproteins. So, I'll show you the diagram. I'll show you the diagram here. Proteins are here. Carbohydrate molecules join in. It is nicely in the diagram. I'll show you. Don't worry. Glycoproteins, right? And other carbohydrates containing molecules secreted by the cells. Most abundant glycoprotein in ECM, right? Extracellular matrix of most animal cells is collagen, which forms strong fibers outside the cell, right? Most abundant collagen fibers, right? I'll show you. The collagen fibers embedded in network woven ethila winding, right? Out of protoglycan, right? Proteoglycan, peptidoglycan, proteoglycan, not peptidoglycan. That means again a chemical, right? Secreted by cells. Functions. forms a protective layer over the cell surface. Yes, you can see protective surface over the cell, right? 3D right? Then cell is a football. Football, right? Cell is a football. Now you are going to cover that football. You're going to cover that football in the football. Right? You're going to cover that football. It's a football. Huh? You are going to cover that football with coil fiber. Look, coil fiber again, football is covered. It is nicely covered. Extracellular matrix is like this. Football is covered. Right? It is like this. So think it in 3D. Yeah? 3D. Linking extracellular matrix and cytoskeleton. Just link around, right? Influence the cell behavior by involving the mechanical and chemical signaling. That you don't learn a lot, right? Mechanical and chemical signaling for some cells to produce antibodies or antibodies produce karana, right? Cell communicate karana, you don't learn about those things, right? Cell communication is important, right? Cell movements, muscle movements, right? Mechanically. So signals, messages are sent in between the cells. Cells communicate. Cells communicate. How the tissue behaves. When the signal is coming, the whole tissue should behave together. So for that, all the cells should communicate. Right? That communication is also done by this extracellular matrix. Right? Influence the cell behavior by involving in mechanical and chemical signaling. Right? Tell me whether you get that right. Right. See, this is the plasma membrane. The plasma membrane, right? Plasma membrane. Very recently we learned. Can you remember? Very recently we learned about the cytoskeleton. In the cytoplasm, this is cytoskeleton, microfilaments, microtubules. The cytoskeleton. We finished that part, right? Here you can see. Take this one. Right? Glycoproteins. This is the protein. The protein, right? Protein. And carbohydrate is bound to this one. Fix these are the units which fix the other fibers to the cell. Fix. Then you can see large collagen fibers. Large collagen fibers. So this is outside. Outside. And this is inside of the cell. This is inside the cell. This is inside the cell. This is outside the cell. This is the margin. Right? These are collagen fibers. Right? Protoglycan, right? Your name. They are not that important. Campbell even this molecule was taken out and described. We don't need that one, right? Right? These were discussed earlier. This fibronectin, right? Collagen fibers, plasma membrane, right? Okay. I hope you have a good idea about this one now. Make Campbell photo. Right. So again, this is the outside. The outside, and this is the inside. This is the margin. Right? This is extracellular matrix, and this is cytoskeleton. This is the cytoplasm. Right. Hope you have a good idea. Alright, finish. Right? With him, we join. Right. Right. Then right? Even when we were learning the old syllabus, we didn't have this one, right? Cell junctions, tight junctions, gap junctions, right? And plasmodesmata. That means, right? The cell, sorry. Okay, mama follow me. Right? Cell junctions. It calling plasmodesmata. Cell wall එකකට තියුටයි මට මේක හොයා ගන්න බැහැ. Right. Plasmodesmata is then until right? Cell junctions, right? Cell junctions means now it is like this, right? In between the cells, then the cells there are some relationships, right? And it's one cell connects to the other in different ways. One cell connect to the other in different ways. Those are commonly called as cell junctions. Sometimes they tightly fit with each other. Tightly fit together, right? Sometimes they fix the cells. And cells fix revert just like riveted lines. Right? Sometimes these junctions help in the transferring of materials. Materials from here to here, here to here, right? So cells have relationships. There are relationships here, right? In between the cells. They are called as cell junctions, right? Mainly 3 types of cell junctions are in animal cells. Okay, animal cells is like, right? The cell junctions are structures at which neighboring plasma membranes are joined. Neighboring plasma membranes, yeah, right? Are joined. They are also interact and communicate via sites of direct physical contacts. Communicate, right? How this happens, right? Functions connect the internal bulb in chemical environment of adjacent cells. Cell junctions are structures. Cell junctions are structures at which cytoplasm of adjoining cells are joined. There are 3 types of junctions. Right? There are 3 types of junctions. Right. First one is called tight junctions. It is like this one here, right? Immediately after epithelial, most of the time, immediately after epithelial, I'm going to grow it very large, right? Epithelial. Imagine this is the epithelial present in the digestive system, right? So it is showing finger-like projections. This is the epithelial cell layer. Right. The next cell layer, let's go for the next cell layer. Now imagine you are going to take a meal, like right? Then water goes like this. Water like this. Then they flow like this. Some of them will absorb. When the liquid goes, it can go through these membranes. In between the gaps of this epithelial cells. Then, if they start to penetrate for the other tissues also, there is a problem. Right? They are absorbed with the water and the essential amounts are sent. But if the water is going to squeeze to the other parts, unnecessary parts will you take a question. It's a problem, right? So to avoid that one, these 2 cells, minimum cells, right? So the next layer, most of the times, the next layer after the epithelial next layer, right? Next layer after epithelial is tightly fitting. Here, these 2 cells, A and B, are tightly fitting with tight junctions, just like glue. Waterproof glue thing, right? I understand, right? Waterproof glue, right? In between this tight junction, in between A and C, again tight junction. So water doesn't get penetrated. Again in skin, on skin, also epithelial is present, right? Skin epithelial, right? When you are washing, and all that, right? Do water get penetrated to the body? Absorb, yes, small amount is going, right? but water, body water level, you know, water bond, right? When you are thirsty, you're going to sink, you're going to bath in water, right? Going to swimming pool and be like, no, water doesn't get absorbed like that in us, right? Because under that one, that's a good thing. Huh? That's a good thing. Right? Under that epithelial layer, there are tight junctions in between the cells, so they do not allow the penetration of water. Right? It's prevented. They are called tight junctions. Tight junctions, right? Connect the plasma membranes of adjacent cells, nearby cells. Adjacent cells tightly bound by specific proteins forming continuous seals. Continuous seals around the cells. Prevent leakages of extracellular fluids through intercellular space. Leakage of extracellular. These are not cellular, these are cellular things. Extracellular. Whatever flowing here, extracellular. Fluids through intercellular spaces. Here, intercellular spaces are present. There are two spaces in between, right? But due to the presence of this one, no. Further, the area which I'm going to say X area from Y, no. Liquid can come up to this point, but from here onwards, no. Doesn't happen, right? Example, skin epithelium, right? And the digestive system is also like that, right? Digestive tract. Next one, right? Is it clear? Right. Right? Desmosomes or anchor junctions mechanically attach the cytoskeletons of inside part, right? Of adjoining cells by intermediate filaments for strong binding. Example, muscle tissue, right? Clear. I hope it is clear, right? Next one, in the muscle tissue to form the tissue, make a Campbell right? You would have visited the right? Muscle in the muscle, imagine this is one cell, right? I'm a branch, right? One muscle. Cylindrical, make a skeletal muscle, right? Draw it like this. Muscle cells, muscle fibers get together and forms the muscle tissue. So for a movement like this, when I'm moving, heavy weight lifting, now running, these things should be moving together. Individually move, right? To move together, right? All of them are tightly bound. How they are tightly bound? They are not glued like this. How this happens? Inside this cytoskeleton is present. That means cytoskeleton, right? Cytoskeleton is present. This cytoskeleton joins with the other cytoskeleton. This cytoskeleton joins with the other cytoskeleton. Like this. By using microfilaments. Microfilaments. We call microfilaments, right? This is what happens. Right? It is not given for you to show, but I hope it is good to show. Right? Is extra. We got I have to teach this later under the motion of cell mechanism of Huxley's mechanism. Teach it. Only lot to make a super slow. You identified stations in meiosis. Second. Right, give me a second. Yes, any question? Any question? Right. I know. I think my option එකක් තියෙනවා මම එදා කරා එක දවසක් A level class එක I don't like to do that, right? If you unmute the microphone, කෙලින්ම යනවා ඔයගොල්ලෝ meeting එකෙන් අයින් වෙලාම then meeting will be locked for you. That means you cannot come into the meeting again, right? පොඩි ළමයින්ට වගේ හරිනේ. Right. This is how it is how it looks like, right? මේ ප්‍රශ්න තියෙන්නේ උදේ වෙච්ච දේ හින්දා නැත්නම් මේ ඔක්කොම images ටික තිබුණා, right? ඒක load වෙන්නේ නැති මොකරි something happened, right? Some of them are there. Some of them are showing this problem, right? Okay. See, now we are talking about these things, right? This is one cell. This is Campbell biology, right? Campbell biology image. The electron microscope. Can you remember Venus creations ki area and the Chutikal? Can you remember when you in O levels? Now, this is right. So this is one cell. Actin, myosin, biotin, right? Imagine this is one cell. They have given the boundary. They have marked the boundary. Right? They have marked the boundary. One cell. And the other two cells, joining two cells, right? Then what happens is these two cells are joined nicely. They are joined by this actin and myosin. Can you remember these microtubules? Or maybe very recently finished. These are actin, not myosin. Forget the myosin, right? Actin filaments. Actin. These two cells are nicely joined. Now these two cells are joined. This is one cell. Okay. One cell, the other cell, right? The other joining cell. Using the green color structures, now we can see cells are joined. Right? By whom? By this one. These people, right? Actin subunits, right? Actin. Is it clear for you? Should be, yeah. The other thing is then tissue tearing a tissue. Muscle tissue. That means these bonds are broken. Maybe you're damaging of this. In the Campbell also term revert reverting. Revert I can use reverse. To join two surfaces, reverse. In between a nail is going. Just exactly similar to that one. Revert karma. Right? Exactly similar to this. Reverting. Revert karma. Right? Revert. Right? Second one. Campbell එක දිනවල ලස්සන එක ඒක විතරක් right ඒක විතරක් මට ලස්සන Campbell එක තිබ්බා it was really beautiful right මට අපෝ කියවන චුට්ටක් tight junctions කියලා Campbell එකේ තියෙන එක I'll read the Campbell one tight junctions the plasma membranes of neighboring cells are very tightly pressed against each other bound together by specific proteins right forming continuous seals around the cell, tight junctions establish a barrier that prevents leakage of extracellular fluid across a layer of epithelial cells. Right? Right. For example, tight junctions between skin cells make us watertight. Next one. right? Desmosomes, also called anchor junctions, function like rivets. Function like rivets. Fastening, fastening cells together into strong sheets. Intermediate filaments made out of keratin proteins anchor other actin, right? Anchor desmosomes in cytoplasm. Desmosomes attach muscle cells to each other in a muscle, right? Some muscle tears, muscle tears involves the rupture of desmosomes. The rupture of desmosomes, right? The muscle tear. This separates. Pain, right? So cannot form. It takes a long time. Even this break, it can form, reform, until then, right? Especially this happens for athletes. Right? Their movements, right? Okay. Next one is gap junction. Gap junction, right? Gap junction is nothing, right? Gap junction means two rooms adjoining next room. Imagine your room is a cell, one cell. Next day, right? Next room is the adjoining one. In between the two cells, there's a wall. You're going to make some holes. The wall, right? The wall, you're going to make some holes. 2, 3, 4, 5, 6 holes. And you can communicate. That means you can take water if you like, water gun, right? Right? You can send materials if you want for the other side. Food can be taken. Actually, your doors and windows are the example, right? But doors and windows are small in number, holes. Likewise, in between the cells, if there are cells could happen, right? Right. In between the cells, materials are shared like this through holes like this, right? Holes like this. And these junctions are called gap junctions. Actually, there are holes, right? At Kiola, they are covered with proteins, right? Gap junctions are right? Also called communicating junctions. Things are shared, right? Provide cytoplasmic channels. Channels means pathways. Cytoplasmic channels means pathways to connect the cytoplasm. Cytoplasmic channels from one cell to an adjacent cell, and in this way are similar in their function. In this way are similar in their function to the plasmodesmata. We didn't discuss COVID, right? Gap junctions consists of membrane proteins that surround a pore through which ions, sugar, amino acids, and other small molecules may pass. So this is not just a hole. Imagine this is the hole. It's the hole, and it is surrounded by proteins. Proteins are surrounded. Right? So across this, ions can transfer. Across this, right? Sodium, potassium, ions can transfer, right? Sugars, amino acids, and other small molecules may pass. Gap junctions are necessary for communication between cells in many types of tissues, such as heart muscles and in animal embryos. Communication, right? It's really important, right? Let's look at another image to describe. Now it describes all the three images. Right. Take this image now, right? First one, tight junctions. Can you see? I'll explain. Tight junctions prevent the leakages by tightly fitting. Can you see this purple color image? Purple color area. These things, see? See, fluids can come like this and flow on this one due to the presence of tight junctions here. Here the cells are tightly packed. Cells are tightly packed. Right? So this extracellular fluid cannot penetrate through this like this. Cannot go like this from here onwards. It is prevented. So this purple color, tight junctions, right? This is the Campbell biology image, bioinvite is present for you, right? This is the electron microscope image, right? Right. Then cells are connected through fibers, see, just like rivers. Two cells, adjoining cells are connected through rivers, rivet curl. Here also something like this. They are called desmosomes. Rivet curl, right? Me ani patra painting the other way. Me kata room me hole ekak. This is the hole, right? I cannot draw it up. This connects to the other way. 3D, think it in 3D. Riveted. Third type, they are not just rivets, right? Inside that there's a hole, small tube, right? They are tubes which connect the two cytoplasms. Tubes. In between those tubes, right? This is for the other side. Across those tubes, materials can transfer. Gap junctions, right? Materials can transfer across these tubes like this. Right. Three types, right? Then let's go for the plasmodesmata. I'll discuss the one which is in our book. Right? This is animal cells, right? Now, plasmodesmata is present in plant cells, right? Plasmodesmata is present in plant cells, like this, right? I'm running out of this black color. I'm really, usually black color I'm very good use, but these days I'm really protecting this one because the refill is also at the bottom, right? Due to this travel restrictions, it's really difficult for me to go there and get the refill here, right? Right. Plant cells are like this. Cell wall is present in between two cells. Cell wall. Now, in all levels, so when we are talking about the cell wall, you may have got this structure. Cell wall, make a mukad. Tell me, yeah, if you can remember, I have taught you the cell wall. This line. Can one make a mukad can be X. I named this one, right? Black, right? One cell wall, the other cell wall, adjoining cell wall. Actually, it is not like this. Again, in between them, I'm asking the exam middle one. Right? In between them also, you can find some connections. Tell me the answer. This X, the layer in between two cell walls. 2 weeks ago, I told you. Here also, you can find channels which connect the two cytoplasms. See, now things can transfer in between this cytoplasm. So I'm getting the answers. Right? From here to here, things can transfer. These are called plasmodesmata. Right? Yeah, middle lamella, very good. I microphone again, calling up what I think. Then our setting microphone time. Yeah. Yes, very good. Middle lamella. Calcium pectate, magnesium pectate, right? Okay, yes, things go like this from the two sides, right? In that one, Campbell, right? Animal cells, this thing is done by gap junctions. Just like plasmodesmata in plant cells, gap cells in animal cells, right? Just like gap cells in animal cells, now we see plasmodesmata in plant cells, right? The difference is this goes through the cell wall. In us, there is no cell wall, right? But same thing. Microscopic channels, very small channels. Channels means pathways which connects to, right? Channels which runs through plant cell walls. That is the specialty. Which runs through plant cell walls. They are cytoplasmic living connections between cytoplasms of adjoining cells. They connect X and Y, two cytoplasms. These are membrane-lined channels filled with cytoplasm. So these are, if you take a cross section of this one, right? Cross section, these are covered with a membrane. Yeah, membrane is covered, right? Not just holes, right? Membrane, right? Okay, go. Okay, right? Filled with cytoplasm, right? So this is filled with cytoplasm. Right? Okay. So in the is it clear for you? Then clear that is the advantage of having an online class, right? Online advantage Google. Google can a project successful. It's not that successful, right? When using a projector and all that, it is not that successful, right? Because that is done in the universities. Smart board in universities, right? Smart board, whiteboard, and a green board. Right? Even that is not successful. I feel this is so successful, so close for you, right? Should be super clear for you, right? Not clear, you know. Right. So in the next class, what we have to discuss is, right? Next class, right? Finished, right? Next class, let's try to finish the thing. So I want you to read this part. Right? Yes, you know about this mitosis and meiosis, but this is totally different, right? Totally different means you are going to learn a lot of things now. A lot of new things are going to be taught here, right? So next topic of us is the cell cycle, right? Cell cycle. I'll tell you what we have to discuss, right? Or G1, G2, growth, synthesis, right? Space and mitosis. Oh yeah, right? Cell cycle means, I'll give you a brief idea, right? You know, cell cycle means the life cycle, cell cycle, right? So just like us, right? Cell also has a nice life cycle. We also have a life cycle. If you put that into a cycle, it's something like this. There is a growth. We have a growth, right? We have never in one, right? Reproductively active age is there for females. Then there is again growth and the rest of the life. Rest of the life. A cycle we cannot exactly say a cycle because it is not cyclic. Actually, at the end, we die. Right? When it comes to a cell, it doesn't happen. At the end, cell theory. New cells are formed from the pre-existing cell. So it goes a cyclic. Yeah, cyclic. Right? New cells are formed from existing cells. So in the cell cycle, it can be divided into 4 main parts. Cell lifetime is divided into 4 main parts like this. First one is called growth phase. Right. G1 actually. Growth. That means the small cell. This is the initial cell which started from the large one. Right? New one. It grows in this growth phase roughly to be a support for the next class. Roughly what happens is organelles large, organelles grow, size of the cell increases likewise, right? Things increase. Things happen, we discuss, right? A lot of things are point-wise, right? Growth things happen. Then there's a phase called synthesis, S1, space, S, right? G, S, right? S means synthesis. New organelles are synthesized, right? Especially DNA materials to be exactly similar. DNA and RNA materials are synthesized. New organelles to be double then divide later. divide kare. New nucleus should be formed, right? Some nearly similar amount of mitochondria, chloroplasts, but we don't talk about the plant cells here. Right? All the other organelles should be synthesized. So in this phase, these synthesized things, right? Synthesized then again they grow. G2. Right? Then everything is finished. This has reached its maximum maturity, maximum growth. There's nothing further to be done. I can run there, right? Maximum level. Then what happens? This divides into 2. This is the cell division. G2 is the division. Cell division. Right? Okay. So we briefly discuss these things. We briefly discuss. And we discuss in detail what happens here. In detail, we discuss right? The cell cycle and the processes of cell division. Right? The sequence of events that take place in the cell from the end of one cell division to the end of the next cell division. End of one cell division method. Right? End of one cell division method. Clock. End of one cell division to the end of the other cell division. End of one cell division. Right. End of one cell division. End of one cell division and end of the other cell division. Right? One cycle. At the end of the cell division, two genetically identical daughter cells resembling the parental cells are produced in mitosis. Right? Eukaryotic cell cycle, mitosis. So done. We have learned a lot. Interphase and mitotic phase, M phase. Right? Interphase is the longer phase of cell division. Right? See? Interphase. Interphase. Long. Right? 80%. Interphase is the longer phase of cell division. It covers about 90%, depending. 90% of the cell cycle. 80% 90% of the cell cycle. Interphase could be divided into 3 phases. Interphase, right? So this is commonly interphase. G1 first gap phase, growth, right? S1 synthetic phase, right? G2. In G1, right? In this phase, synthesis of proteins and production of cellular organelles leading to cell growth occurs. Growth occurs, right? Proteins essential for S phase are produced during this phase, right? Growth occurring, right? S synthesis. What is synthesizing? Okay, especially DNA and RNA. Exactly similar genetical information should be there. For that, exactly similar copies of DNA should be produced, right? DNA replication, we are going to discuss, right? Occurs and synthesis of histone proteins take place. DNA wind around the histone and binds from chromatin. G2, cells continue to grow. Second growth. Right? Second growth. Cells continue to grow through proteins, through protein synthesis as well as cellular organelles. Right? Proteins essential for mitotic phase will be synthesized. Duplications of centrosomes take place. There are cell cycle controlling checkpoints available at G1, G2, and M phase to ensure that the cell is ready for moving into upcoming phases. Of cell division. Some cells receive a go-ahead signal at the G1 checkpoint. It will usually complete G1, S, G2, and M phase and divide. If it does not receive a go-ahead signal at the point it may exit the sales cycle, entering into non-dividing stage called go phase, right? The most cells of the human body are actually in the go phase. Example, nerve cells and muscle cells. Remember, they are going down. Why the new cells should be produced? Cells, right? This cell cycle doesn't continue. There are trigger points. Right? Right? Trigger points. If this happens, signal right? I am finished. You can start. I am finished. You can start. I am finished. You can. Relay relay can baton pass relay running, right? One finishes his job and pass the baton. One finishes his job and pass the baton. First one ran, pass the baton to the next one, right? Trigger point is passing the baton. Second one also running and passing the baton. Third one is also running and third one doesn't pass the baton for the fourth one, throws the baton. Then can the fourth one not? No. The rules of relay, you should have the baton with you. Baton is the end, right? Baton should be with the person who is running. Otherwise, it is not valid. Likewise, if the third person throws the baton, right? Then what happens? This cannot go for this space. Fourth one cannot run. Likewise, some cells do not undergo cell division. Remember nerve cells, especially. That's why paralysis and all things can happen. If a nerve cell is damaged, that's why they are innermost cells. Right? You cannot repair. That's why the brain damage, brain damage, right? Brain damage, brain cells, if they're damaged, they are also nerve cells, right? They cannot be recovered. Muscle cells, it's a different story, but some muscle cells, right, also shows the same thing, right? Blood cells we don't take them here because there are no organelles and all that. Blood cells is red blood cells lifetime. Again, new cells are not formed. Again, the bone marrow produces new cells. Like a sort of stories are there, but what you want to know is some cells undergo this cycle and some cells do not go this cycle. They go for go phase. Right? Remember that incident, throwing the baton on the relay, right? Right. So that's it. That's enough for today, right? Because next one is mitotic phase and all this prophase, anaphase, metaphase, right? Terms comes into play. First read it. Next week, no problem. Small thing, but please read it in comparison with the Campbell biology, if possible. Videos, then it will be easy for me. Easy for me, right? Then energy relationships. It was make you right. At the end of this month, okay, can finish the first lesson. Right. So with that part, I finished today's session, right? Yeah, pull. Yeah, rewatching the videos, I can share them privately. Group send a message to the group. You want to rewatch it. All the things share after 3 more months, all the videos 3 hours, right? 3 hours into sessions 60 hours, right? If you want any session or anything, you can send me the email. I will send it to you privately, share it to you, right? If you want, no problem. Call email, right? I want this lesson, no, that part. If you want all of them again and again, I will share it to you. No problem, right? Impression useless. Right? But I think note right? Rough idea, you have the rough idea, right? Look at the note in the papers and tutorial discussion. Again, we revisit these things. But if you really want, or if you miss any class, obviously request me. Right? Anytime, at least give me a call. Right? Somehow you can get all the videos, no problem, right? One was not recorded. The YouTube software issues. Live streaming. I forgot time this, yeah. Forgot to put it, right? Class YouTube private share, right? YouTube. Right? It is shared in private. Right? Okay. If you have any doubt, ask me with the email. Right? My email teaching first. They will send you the email, the videos. Right? Okay. Right? Looks like no questions for today. Right? Okay. Then. All right. Okay. I will allow you. Oh, you don't want to hear. Right? I can allow you to record from there. Right. Okay. Then I'm going to wind up the session here. Right. Okay. Then Google make right? Google 10 to 15 even make it's going to grow up. Again, if you have friends, then in right? It will be really a problem now. Advertise then right? So Google ending contact right? කොහොමදත් මේක අපි 100 යනවා කොහොමදත් right මේ අවුරුද්දේ results එන්න කලින් ඊට පස්සේ අනිත් group එකත් එනකොට ඔක 250 nothing right ඒක මම ඔය සද්ද බදලේ තියෙන අනිත් class මෙහෙම අඩු වෙනවනේ දැන් එන ළමයි කොහේ හරි class ගිහිල්ලා නෙන්නේ එහෙම තමයි වෙන්නේ right slows down it goes up right? This goes up okay then if you have any question you can ask me by any means right okay then thank you very much read this up so it's easy for me and you both right thank you very much and have a good day. All right, okay. I'll put some posts. Mandana post right? Share right? I'll put some post. Right. I'll put it to the group so you can share it. Right?